Hey guys, so let's go through the last, not the last two, the next two sections in chapter 11. So we are going to talk about the mechanisms. How do we get things to evolve? How does that happen? So it starts off mentioning Hardy-Weinberg equilibrium, and that would be if everything was perfect, nothing ever changed, there was never any pressure, selective pressure, then the frequency of those alleles would always remain the same. We would always see the same level of brown eyes and green eyes and blue eyes. Those would always stay the same. However, nothing stays the same in life ever. So we have fluctuations. Some of those fluctuations are due to natural selection. So we mentioned natural selection before with the founder species of those finches that had blown in from the mainland and they landed in Galapagos. And then it was just like, holy cow, look at all of these resources for me to eat, all these different places that I can nest in and survive in. And eventually, over time, the variation in the finch offspring served as adaptations, and they eventually underwent speciation and branched off into all of these different environments, and they changed into nectar eaters and seed eaters and insectivores over time. So these gradual changes in a population over time. Again, it is not like Lamarck's inherited traits that happen on an individual. An individual does not evolve. A population evolves, not an individual. So with natural selection, that is another method of um, changing genes and causing pressure to be an exception to Harvey Weinberg. Mutations are the ultimate source of variation, like we learned about in the last chapter. What are we talking about when we talk about mutations in genetic makeups? Well, there's a couple different types of them. There are a few. Let's at least look at them and go through them. So you have your gene code, and we talked about when we undergo cellular respiration that we're going to copy that DNA and we're going to make a copy, a total carbon copy of it. But sometimes things go wrong, and so we have checkpoints, right? We had checkpoints to come along and make sure that that DNA was okay and correct any errors, hopefully, any mutations. But if we didn't, then a mutation may crop up and it happens. So here's some different types of them. So this first one is called silent, the silent mutation. So we are looking and um, we have DNA, we have messenger RNA, and we have amino acids at the end. So this is central dogma straight up that we have learned about over the course of this semester. So here is our central dogma, and we are going to have our DNA, and we have A and C and G and T, A and G, A and G, C and G. That's the same. T is the same. This should be an A, and it's a T. That's not right. Um, that's not right. That's a mutation. So the T is stuck there where there used to be an A. We have a mutation. Oh no, what are we gonna do about the mutation? So with the mutation, what's it gonna code for? Is it going to affect our end result, which is going to be the amino acids, the proteins? So let's see, when we put this in, and we are going to use this just like we did with the plasmid project, we have our amino acid table and we plug and chug. What did we code for? So here was our template DNA. Here is the RNA that we coded for. Here's our messenger RNA codon, GUU. And GUU still codes for the same valine. It still codes for that same amino acid. Cool. We have a mutation and it didn't show up. So it's silent. This one is silent because it didn't change the protein. We're still going to express these genes and get the same end result. We still get the same protein, which is awesome. So this is a silent mutation. Even though the genetic material changed in the DNA, we still got the same result. So it's okay. What about a missense? What about a missense? So with missense, we are going to have, where's our codon? GTA. Where's our next codon? Our next one is CCC. This codon is ACC. So this is wrong. We got an A here where it should have been a C. Let's see if it matters. Let's see if it matters. So if we have an A on there. Are we going to change up our genetic material? That that isn't what it should be. It's not what it should be. So our codon or messenger RNA says ACC. Not right, but whatever. We're going to move on. Because what I really want you to determine is our codon is wrong. Our codon is wrong. This really should be UCC. But regardless, it's going to be a different amino acid, and this is the end game. We have a different amino acid. So this is totally a mutation that shows up in central dogma. It's going to show up and give us a different protein. And all it takes is one amino acid switch, guys. If y'all have ever heard of sickle cell disease, that is literally one amino acid switch. It is as simple as something just like this, a missense in the DNA when it was being copied over, when it was undergoing replication. One little chain, one little nucleotide that swapped out in the DNA makes a change in one amino acid, and that changes up hemoglobin, and that causes sickle cell disease. So it could be one simple little missense like this that shows up and it has a huge impact on an organism. So this we've talked about silent, we talked about missense. Let's talk about nonsense. It has no in the word, and that's going to help us remember because at the end of nonsense, here's what we end up with. We have AG, and we are going to end up with UAG mRNA, which it UAG. There we go. Because that should be ACC. This one is UAG. And if we have UAG, if you remember, we had a few different ones of these. There were 3 total codons that could potentially end up with the same process, and the end result is stopping it. There are 3 stop codons, and UAG is one of them. So if this one is accidentally coding for UAG, that means it's going to stop. And so our protein may be completely shorted. Maybe this was just like the 4th amino acid in a strand of 100 amino acids put together for this really long protein. And we only got 4 out of the 100 amino acids because we put in a stop codon in there. And we stop, it just stops going. We don't have any more. The ribosome's just going to stop adding in the transfers and it's done. It thinks it's done. So this is, excuse me, this is nonsense. It has no in the name. It doesn't go anymore. It goes no further. There is no more translation of this if it accidentally codes in a stop codon instead. So this is nonsense. It means no more. And then other types of mutations, insertion and deletions. Insertions and deletions. Insertion. Let's look at our letters. We have A, G, C, G, A, G, C, G. Everything's good. What happened? We had CCC over here, and here is CCC. We inserted 2 random nucleotides, got stuck in here and inserted in there, and that throws off everything. Everything. It throws off everything. If we remove those guys, look at what happens. These are the amino acids that we code for without the T and the A. And if we have the T and the A that got inserted in there, these are the amino acids that we code for. So insertions and deletions affect the entire protein that you are going to create from that point forward. So these are insertions and deletions. They are a big deal. They completely shift the reading frame on these guys. So these are different types of mutations. They lead to changes in the protein sequence, unless we're talking about silence, and silence don't lead to changes. Genetic drift is another thing that we need to talk about. So with genetic drift, that's going to be a change in allele frequency. So in this case, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 10 bunnies. We got 10 bunnies. And 8 out of the 10 bunnies are brown. 8 out of 10 are brown and 2 are white. We got a 20% chance of some white bunnies. And on this second one, oh gosh, the dogs are going crazy. On this second picture over here, we now have shifted. Our bunnies have moved. We no longer have a 20% chance of white bunnies. We now have just one lone little white bunny over here in the second generation. So now we're down to a 10% chance of white bunnies. And now we're going to have reproduction again. And by the third generation in the F, this would be F1, and here we are at F2. By the F2 generation, there's no bunnies that are white. Everybody's brown. So we have lost a bit of allele there. We've lost a trait where we don't have the white bunny showing up anymore with this genetic drift. We're changing our frequency, and it is based solely on chance. That's what we're assuming that this genetic drift is. It's based solely on chances when this happens. Sometimes genetic drift gets a helping hand, in usually a negative way. So usually it's a natural disaster. Sometimes it's an artificial event. Um, but some sort of disaster befalls a population of animals. So I like to think of this as a national disaster came along, like maybe remember last year the fires in Australia were terrible, and forest fires were everywhere in Australia, and they were burning up everything, and the poor little koalas were severely impacted by that. That's a bottleneck effect. Acres and acres, hundreds of acres were burned, and their environment was completely changed, and we lost a lot of animals, so many organisms in that event. That event has now limited us. Here we have red ones, and we have green ones, and we have orange ones. Now we're down to just orange ones and green ones. They're the only survivors that have happened. And maybe that bottleneck effect is people hunting things, because we used to really like the dodo birds, and those were delicious, and they couldn't really run around. So we're going to eat those things because they taste like chicken, and we ate them all down to like two. That would be a bottleneck effect. It's where something is killing off a large portion of the population. Typically, we're talking about a natural disaster um, that is just randomly killing off the birds and that, or whatever it is, the koalas. And we end up with a bottleneck effect where we had a huge, huge genetic gene pool. Remember, gene pools are great. The bigger, the better. And then we end up with this teeny tiny shallow gene pool, and that's not what we want. And that is bottleneck effect, where a natural disaster or something like that can influence and make genetic drift even worse. We talked about founder effect, gene flow. With gene flow, this was where individuals are just going to flow in and out of the population. So in this case, we have a brown beetle, and it's just going to go and check out and see what the green beetles are doing. It just wants to know what's the green beetles doing over here. So it comes and hangs out with its buddies, the green beetles. And maybe a green beetle wants to check out and see what the brown beetles are doing, and then it'll go back. So it's how the different alleles flow into and out of the population. Maybe it's beetles, maybe it's deer, maybe it's a cougar go into a different population in a different area of a giant territory. This is gene flow, where they migrate into and out of different populations. All right, so that's the end of section 2. Section 3, evidence of evolution. Theory's great and all, but how do we prove this? How do we know that evolution really happened? I mean, come on, I could have just made this stuff up. I could have made this up. How do I know that I'm not just lying through my teeth? Scientific discovery, scientific method, testing and testing and testing, evidence based. It's all about the evidence. So where's the evidence? Where is the evidence that evolution really happened? Because maybe I'm just yanking your chain. The fossil record has tons and tons of evidence. We can, dig up these guys and find evidence that things have changed over time. So we see all sorts of fossils, all sorts of things embedded in the rocks, going back thousands, again, and hundreds of thousands of years out there in the fossil record. So that is one of the great uh, sources of proof for this is in the fossil record. What about this homologous and analogous? Let's go over that just one more time. Remember, with homo means the same. So everybody has the same trait. And why is it the same? With homologous, it's the same because the common ancestor had it first. Common ancestor had it, and then everybody else had it. So originally, all these guys are tetrapods, the first amphibians that crawled up out of the water. And then eventually our amphibians turned into reptiles and birds, and eventually we're going to have mammals out there. But all of these guys hail back to the original tetrapods, and we all had the same basic structure. So if I asked you, when we compare a bat wing to a person's arm, those are homologous structures, right? Here's a human, and you don't really see a bat wing's bones, but it still has the same thing, except for its wing is its finger bones really long out there. It's really fascinating to check out. But those are an example of homologous because the common ancestor had those arm bones. We all did. But what if I asked you about a bat wing and an insect wing? That's not the same. So it's both the wings, they're both wings, but the the common ancestor didn't have wings. There's not a common ancestor link between insects and humans that has wings. So that doesn't fly. It doesn't work. So if you are confused about this, homologous the same. The common ancestor had it. Analogous may be a similar trait like a wing or white coloring, but it did not come from the common ancestor. That evolved separately, like convergent evolution. So take this quiz. Oh man, I bet this doesn't work anymore because it uses Flash. Let me know if you have questions and you're concerned about the difference between homologous and analogous. There will be 2 questions on the test on it. I know there will. Let me know if you have concerns about this. Ooh, another vocabulary term that we haven't talked about, vestigial. Vestigial. It's a structure that the common ancestor had, and we said, small changes over time may change an animal's body. And if it's a structure that an animal hasn't been using for a while, it is vestigial. Vestigial. So snakes are reptiles, and reptiles were tetrapods. They are tetrapods. They have 4 limbs, but snakes evolved away from that. But they still have the limb buds where those lower limbs went, where their limbs went. So we see little limb buds, little legs, little baby limb buds where their legs should be on the snake. And some of them you'll be able to see these things. Some of them you can't see these things, and that you won't find them. But if you see a structure like that, it's a vestigial structure. It's something that is not used. Here's an example again of an analogous structure. They're both white, but that's not because the common ancestor was white. That is convergent evolution, and it is an analogous structure. It's an analogous structure. Another term we need to know, biogeography. So we are going to grasp that evolution happened and it just changes over time in a population of organisms. And we need to also keep in mind with biogeography that the land has changed over time too. So not only have the organisms changed over time, but the land has changed too, because originally it was Pangea that we all learned about, right? We had Pangea and all the continents were one big supercontinent, and then it broke up into 2, and then it broke up and it broke up and it moved and moved, and the tectonic plates have moved everything everywhere. And so these things that were originally all slammed up next to each other are now far, far away. So we need to take that into account when we look at these things and evolution and how in the world did something from South Africa get all the way up into India, how did that happen? Well, remember that all these things were smushed together. So that is biogeography. And um, even the molecules, if you were a molecular biologist, even our genetics, even our DNA is ancient. That is something that is our universal molecule that we find, and all of the things that we consider alive have DNA. So that is very much one of those foundational common aspects, that everything alive has DNA. All right, so speciation, I'm going to leave for another video.